Good evening, everyone. My name is Chris. I'll be your host this evening for Taxonomy Tuesday, uh, December 9th. Um, if anybody has anything they'd like to share from their computer, please put it in the chat or shout it out. Uh, Hunter sent in some. You want to get us started, Hunter?
Okay. Yeah, so I didn't even attend last week because I didn't have anything and I was getting ready to go to Louisiana and for a foray with GSMS. And so I just finished that up and now I'm in Mississippi and I've got some finds from there. This one is one of the more interesting ones. I have no idea if what it's labeled, is correct. It's just came the from INAT. I mean, I guess that's correct because it really doesn't say anything. But the next picture is cool. If you zoom in more and still more, if that's at all possible, yeah, then you can see. So there's kind of three cool things to look at. And I see Jason's here, and maybe he'd want to talk about what's on what we've got here because I think it's pretty cool.
Might have four.
Yeah, really, at
Yeah,
least.
so from right to left, that thing with the black cilia, the Folios one with the black eyelash things around the margin is going to be a Parmotrema, something in the genus Parmotrema. Behind it, that branchy looking thing is an Usnea.
And
Um,
both lichens, right?
yeah, I see at least five lichens.
Oh,
There's
cool.
six
Cool.
lichens. There's Graphis, scripta all the way over on the right with the,
Oh,
yeah, over there.
mm,
The next one with the larger disc with the broader white apithecia around the margin
mm.
uh, looks a lot a like an in the genus um, Ochrolechia,
Mm.
which tends to have these big apithecia like that. That's one that I would shine a UV light on and see if it fluoresces a different
Oh,
color. I think
and is
you know
that
that.
can you spell that one?
Yeah, Ochrolechia.
I think.
And then next to it, the one with the smaller apithecia and the that are still tan, but they have finer rims.
Yeah.
Uh, I think that's probably my first guess on that is a Lecanora. You can see how it's different than the one with the single larger apithecia to the right.
Mm-hmm.
Um, Not only is the apithecia smaller and the rims are not as wide, but the color of the thallus is different.
Yes. But there are more of the large ones. Would you say on the bottom right or no? Is there just the one of the really large one?
I only see one of the and yeah, that one little tiny one to the left.
Okay.
So that's what I think looks like an Ochrolechia.
Okay.
Um, and then maybe a Lecanora below it, and then what's probably something with that those bright red apithecia. That's probably something in the genus Ramboldia, which is a genus that's much more common down south.
Cool.
I,
Cool.
I think those red things might be Penniphora, a fungus that often accompanies lichen on branches. I put in the chat
Yeah.
Penniphora
Yeah.
rufa.
Let's see.
See if it looks right.
Mm,
Jason, I don't know the lichen that you mentioned, so maybe maybe it's that, but it looks like Penniphora rufa. I find it in the
Penniphora
winter.
Penniphora rufa looks kind of crinkled. They look like little miniature brains.
They're a
These,
little bit
these,
wrinkled. Yeah, they're a little bit wrinkled. So this is not that wrinkled. Let's
yeah, this
see.
one looks a little bit more flat. Um, let me see.
I've got a Penniphora rufa on observation on Mushroom Observer. Let me.
Let's see, Ramboldia. Ramboldia russula. That looks like a common Ramboldia.
That was the most a lot we had. 1.2 million, you think, World War II. Okay, Rick, you're shaking your head, yes?
Yeah, I still think it's a Ramboldia with the kind of light greenish cracked areolet thallus underneath it, which matches on the right. I think those are apothecia on a lichen, not something saprobic growing on top of a fertile lichen thallus. If you look at the color of the thallus on that side, it matches the color of the thallus over there. In fact, you got three lichens on that side. You can see those black lines separating three individuals, same species.
Nearly 10 million.
Oh,
200,000 Pennsylvania men and women served in World War II. Nearly twice as many served in World War One.
cool.
That was surpassed only by
Oh, you got quite the stick there, Hunter.
It is really something. Yeah. Hmm. Yeah. Yeah. So that was cool. Thank you so much, Jason, uh for helping me out. So this is a stinkhorn. And if you focus in really close, you can see these tiny little flies. Those are little, yeah, all little flies. Yeah, wow.
Oh, yeah, you got some good ones there.
Yeah, and they're really, really tiny. Um, and yeah, so these were there. I saw so many. I couldn't believe how many, and they're really bloated. They got a lot of rain, and so they're huge and just so many. So it's I think it's Clathrus columnatus. Um, and it was just so fun to see them, and they do smell. I actually, when I came to Mississippi today, I could smell it. I smell one in the yard, and I'm not sure I found the one I smelled, but I did find another one. So they really come out in numbers down here, and it's pretty interesting. I
Did the top of these ones break away like those other ones?
No, not you. None of the ones I saw were broken. I know what you're saying. I think they generally will stay together. I mean, they are fragile, but if you leave it alone, um, I don't think they necessarily do break away. I'll show another one next week or some more. But yeah, none of them were broken. They all were intact. So pretty cool. And there were other stinkhorns too. But those didn't fare as well with the rain.
How warm was it?
It was cold. Actually, it was rainy and cool. This morning it was 32, but then well, but yesterday it went up to like 64. But over the weekend for the foray, it was in the 50s, low 50s, and it rained at night, like every night. So it was really wet, but it wasn't cold. It just wasn't warm. It definitely wasn't warm, but it was good for weather. But it's gonna get better. The next few days with the sun and it's getting warmer, we're gonna be in the mushrooms. So I should have some good finds for next week. Oh yeah. So Dacrymyces spathularia. I'm pretty sure it looks like a spatula to me, and so I think that's what it has to be. These really had a nice form to them. I don't know if I've seen them looking so much like a little spatula. But these were cool to find. I think, yeah,
Is this picture right side up?
this is how they were hanging. It is right side up. They were, yeah, they were hanging on the underside of a log, basically, that was sort of not laying down flat. It was, part of a log. So yeah, they were on the underside.
I think that log may have been rolled at some point because I think these usually grow upward.
That's an interesting thought.
Also, it's I think that I'm not sure if the genus has changed to Dacrymyces, but at least for a while it was Dacryopenax.
Oh yeah, to Cryopinax, I say yeah. Yeah, I think it's Dacrymyces now. But that's just INAT. I don't know.
Yeah, they're pretty up to date. Michael Quebec still has it as Dacryopenax. And it might be a sort of a gray area also.
Yes, I could see that. Yeah. Yeah, I like the brown the Cryopinax, that one a lot. But so yeah, I mean there should be a yellow one too, but I don't know. I guess they've moved it. Somebody. So yeah, not much more to say about it, unfortunately. It's yeah, and this one I learned when I was out in New Jersey with you guys. So I recognized it and pretty sure I got the ID right. And I think it's pretty common, but just missed or ignored. But it was cool to see and kind of get the name down.
I think somebody showed this last week. Was it Lila? Showed this species?
She's the one who taught it to me. So
okay.
When I was out there, but yeah, maybe she found some too.
Yeah, if I'd seen this, I probably walked past it and have not looked at it. It's pretty interesting.
Yeah, well, she is the one who taught it to me, so it is. Yeah, so it's nice that it's kind of a new one for somebody else too. Yeah. Yeah, I think it's really interesting. And it's just easily missed. It's kind of grows on the almost the underside of a log, it seems. So you could just walk right by it. It doesn't, it appears to be sticking out more than it really does.
It looks like it's got the underside of that thing is just like pretty smooth, huh? No real pores.
Yep,
Yeah.
yeah, we checked that out last week on Lila's, and
Right.
yes, it's like it's sort of like it's steroid. It's like sterium. It's smooth.
Yeah.
The sort of surface is smooth.
I think when it dries, it's a little bit felty. It's a little soft because I happened to find a piece in my backpack today, and it had kind of a soft felt feeling to it. Yeah, so that was good. And so I guess there's a, um 3 or 4 species in Xylobolus. Is that right? I know there's the frustulatus, and then I think there's another couple, but it'd be nice to know all those because I think it's a very small genus.
Cool.
Yeah.
Yeah, I would definitely look at something like that and another sterium.
Yeah, I see three species in the Xylobolus genus for Eastern North America, but one only has 2 records, spectabolus. So it's
Oh.
subpileatus, mostly subpileatus and frustulatus.
Oh, okay. Where are the records for that one? That
Let's see, they are in the Smokies near Asheville, and then one in the middle of South Carolina.
huh.
Yeah,
I'll have to look at that. It'd be
yeah.
fun to find it. Yeah, it's nice to
Ne
be to know a whole
neither
genus, you know?
neither of them are research grades, so they may not be ID properly.
Okay. Interesting. I'll have to look at that one. So this is Lentinellus cochleatus, and they don't find this much down there. And I don't find it much either. But I have found it a few times, and so I think it's one of these things that it's it's common, but it's not often. Um, uh, so, but it's has decurrent gills that go pretty much all the way down and tends to grow in these clusters, and then sort of has a funnel shaped cap. Um, and that's, I think, about all I can say about it, but there's more to say. But I don't know. Does anyone have anything to add? Oh, they have serrated gills, I believe.
They look very oystery.
Yeah, they grow from the ground though. I mean, I've found them only exclusively on the ground personally. Um, and they may be growing on decayed wood, but I've only found them appearing to be terrestrial. And yeah, they do. But they're not they're definitely what I said they are. I'm certain of that.
Yeah, I agree. That's what those are. Yeah, that that long tapering stem with the gills going down it. It's pretty distinctive, and the clusters. I don't find this very often, but it's pretty distinctive. When you find it, you know it.
Yeah. Yeah,
I guess the one thing that a person might confuse would be Hohenbuehelia pelloides. That's somewhat similar, but it has really close, shallow gills
mm.
that are not serrated, don't have serrated edges.
Oh, okay. Okay.
It's a little bit similar. It's usually maybe they're shaped more like shoehorns, but they
Mm.
grow in the same way, sort of on the ground from wood that's partially buried, usually. So there's some similarities there.
Are each of these like individuals like funnily shaped? And
Yeah,
it's a cluster like that. They're all next to each other.
right. Yeah, you can separate them.
Cool.
Yeah. But it's funny, that came up with another mushroom, like, well, a interesting mushroom, but yeah, I mean, is it an individual? Are they individuals or not? Who knows? Um,
I
yeah.
mean, that would just be like cesspitose, right?
Mm-hmm. Yeah, I think so. yeah.
Do they smell like this? Is that the reason?
Not that you would think so. But I smelled them and I didn't get that. So I don't know.
Hmm.
We actually had someone, our main speaker was a guy from University of Tennessee, Knoxville, who is a professor on and teaches uh I'm not sure if it's like flavor science or off olfactory um science or he had been in the flavor business and worked for Mars Candy for a long, long time, and then became a professor. But he talked a lot about smells, and someone said this one was supposed to have a special smell, but I didn't. He didn't say anything about it, and um, so I don't know, but it was interesting. It wasn't what I thought it would be, but it was an interesting talk.
Cool.
Yeah, yeah, the smells are so interesting.
That, that one? Hmm.
Oops.
All right.
I think there's a couple more still.
Yeah?
Yeah. Unless I repeated. Yeah, there should be two more.
Uh, no,
No, we didn't because we didn't go through five, did we?
we did that one.
We did that one. Yeah. At least one more. We did that one.
Did that one.
Oh, hmm. What's the bottom one?
Yeah, I don't think there's no more in there.
Oh, the bottom one, did that open? Mm.
Yeah, that's that one.
Oh, okay. I'm sorry. Well, I'll have more next week then. And if we run out, I can maybe post them up for more up. So thank you, everyone. Thanks for looking.
Thank you.
My pleasure.
I'm jealous of being in a place where there's mushrooms.
Yeah, I'm very, very fortunate and it feels so much better. I was kind of suffering at home. So yeah, it's really good.
Well, does anybody have anything they'd like to share from their machine? I think I have just one if I could. Logged out of it, the iNet. Here we go. Well, this is the little truffle part that I dug up with its friend. I think this is what the iNet came up with.
Is is there a genus attached to that? Because a lot of times INET uses these identification names that consist of a section or a subsection, but they don't. But it doesn't say the genus.
No, that's just what it spit out. I actually have a little book that has the
Yeah, if you click on it, click on the observation, and then scroll down. It says a member of the genus
oh
Elaphomyces, right?
yeah.
Yeah.
Yeah, that's what's in my little book.
Okay.
But this is with the Tolypocladium I found. There's a whole bunch of them.
Cool.
And it's not even
Neat.
in this picture, but there was a couple bigger ones after I took this picture, like hiding in the back. So you dig one up as you do.
I was surprised I didn't see any down here yet. I hope I get to. And you can eat those truffles, right? Deer
I don't think
truffle.
so. Squirrels eat them.
Oh,
Maybe
What?
deer eat them.
okay.
I don't think they're recommended for people. I'm not sure though. I don't know actually.
I think you might want them before the Tolypocladium gets them.
Mm,
Yeah, you would want them before because they're decaying at that point.
uh, that's a good shot there, Chris.
Yeah, I forget what they call these. There was a great talk on NAMA's site about cordyceps just last week.
Mm.
This guy gave a talk. I listened to it twice, and I don't know if I'm any smarter, but he really covers a lot of ground.
Oh, that's right. I have to renew my NAMA. I forgot.
This is all the little sacs that the asci and then thusly the spores are all in there.
But this isn't a cordyceps, right? Or is it?
It used to be. It used to be cordyceps like 20 years, 20, 30 years ago.
Okay, but it like technically isn't now,
Not
right?
it's not cordyceps anymore. Cordyceps was a genus that was assigned to a lot of different parasitic fungi that are ascomycetes. Um, and cordyceps has been quite split apart over the past, say, 20 to 40 years.
So it did and still does include more than those that grow on insects.
I don't think so. I think cordyceps is limited to parasites on insects, and I think there are some other genera that also parasitize insects that probably used to be put into cordyceps.
Okay.
So there's like 3 or 4 species that look like this guy: capitatum, longisegmentum. I think there's one that starts with F. Um,
Yeah, there's a bunch of Tolypocladiums.
oh yeah, well there's a bunch of them, but the ones that have these wide, shiny heads um, on a relatively thick stipe. A lot of the other ones have really thin stipes and heads that are kind of paddle shaped, sort of. They're not rotund. They're sort of thin, in one direction.
These are some more of those sacs from the edge.
Yeah, there's a word for those. What's the word? I forget it.
I don't know. I heard it twice and read it, and I got nothing.
Those are really cool looking though, really neat to see those structures.
Yeah, and to think that all that stuff is loaded up in there so it could get rocketed out.
So is that oh,
Oh, go
so that's
ahead.
microscopy now? Yeah. So what is this now here?
This this is a bunch of spores, I think.
Hmm, cool.
And these are all broken off chunks.
Oh, those are nice.
And then these are still stuck together here.
Mm.
This is the tip of an ascus.
Oh, so when those spores were stuck together, they were inside one of those?
Yeah.
Mm-hmm. Wow, that's pretty crazy.
Yeah, here's the tip of one, and then this is another one that's in closer with the spores still in it, like that.
Oh, okay. Hmm. How interesting.
These are
So
all
those are
broken.
loose spores. Yeah. Hmm.
Then
So
I
do you
measured a few.
you have measurements? How long were the longer ones?
I was coming up with like 43 and a half,
Right.
which I was looking in Mushroom Expert and they didn't really have anything in there.
That puts it in longisegmentum territory, which is 40 to 65. If it's capitatum, it would be 18 to 27.
Oh, that's a big difference.
Yeah. Here, there's a key in an article online. I'll put it in the chat. Um, that has a comprehensive review of all the Tolypocladiums.
Cool. Thank you.
But going through the key, I mean, I just picked a couple that have longisegmentum and capitatum because I know that those are the two that this is likely to be based on the region. But if you go to where the couplet separates, it's let's see, the fertile part cortex composed of pseudoparenchymus peridial layer with an ectal layer, or without an ectal layer. I'm not sure what an ectal layer is. So I'm
Yeah.
going to research.
So cool.
But yeah, when I was out there, there was still a bunch of stuff around. I mean, some of it had looked like it'd been frozen a time or two, but you know, not completely devoid of stuff yet.
Good. Glad to hear it. I mean, we've been, I mean, we're covered in snow. I mean, a couple, like over a foot, like lots of snow still. You guys don't have that?
I think Dave might have some snow.
Yeah, we got between 4 and 5 inches last Tuesday, and we're supposed to have another well, it's variable 1, 2, 3, maybe 6 inches in the mountains tomorrow. It's been very cold here. It was 1 degree above zero this morning
But
at my house.
yeah, same as Illinois. Yeah, really
Yeah.
cold. Yeah. Well, thank goodness, there's still stuff in New Jersey. So glad to hear it. I did see some pictures of frozen mushrooms on the Facebook page, but there was no snow. So that's good.
Yeah, the other Tolypocladium is that looks like the third one that looks like those is let me see, I'm gonna get it now. Uh Valiform, but that one has very small spore segments. Um yeah, I think I agree with Jason. The longisegmentum, according to what I'm seeing on Quebec, seems to fit your microscopy for these spores. they look right and they're the right measurement range.
Funny, that's the first thing that iNet spit out, and I was like, well, I don't know about that.
Oh, it spit out longisegmentum.
Yeah.
Yeah. Now some people say that you can tell that species from capitatum because it's shinier and darker. The head is shinier and darker. I'm not sure how reliable that is. That may have been what iNet was seeing. Also, the pimply surface on the cap, that might be a useful feature also. I'm not sure. Let me
That's
see
got a
what
fancy name
Logo
too.
Codex says.
Yes. Film title 800. 1997. George Clooney and Chris O'Donnell.
Does anybody else have anything?
Well, Batman and Robin. Will? Film title 1600. 2018. Will Ferrell and John C. Reilly. Yeah.
Nothing in the mail.
1200. The islands of the Seychelles are spread over hundreds of thousands of square miles in this ocean.
I've got a few I can share.
Indian Ocean. That's correct.
Go for it.
Losers for 400.
Good.
Oz was known for its concerti featuring this instrument and was considered its leading virtuoso of his time.
Dave, you got an open mic.
Islands for 16. Answer. Available for you.
All right, so a few lichens. This is Rhizoplaca subdiscrepans growing on rock in northern New Jersey. This is non-calcareous rock. I typically see this species in open sun, in kind of rocky outcrops or mountain summit type habitats. It usually doesn't do well in shade and can tolerate the open exposed conditions of these types of habitats. And it's super distinctive, easy to identify. It's got these pale yellow orange apothecia growing right on rocks. Don't have a good scale for size, but this section right here is probably an inch from top to bottom. So it's not a rare species in New Jersey, but it's also not common. So it's always a good find, pretty good indicator of habitat quality, I think.
What's the geographic range of this? Is this coastal or is it more inland?
More inland.
Okay.
Doesn't look like it gets up into northern Pennsylvania for some reason, but it does go up into New England and Canada.
Yeah, because I showed it to Karen. She says she thinks she never found this. Rhizoplaca subdiscrepans. Okay.
Kind of curious why it's not in Pennsylvania more, at least on iNaturalist. We always say that there are three lichen growth forms: crustose, foliose, and fruticose. And this one's kind of, it's not crustose because you can separate it from the substrate. It's not foliose because it doesn't have a clear upper and lower surface. It's not flat. And I guess I guess it would fall into the fruticose category, but technically, if you go to the description, it's called a thallus that's crustose placodioid to subsquamulose umbilicate. So not all lichens fall neatly into one of those three growth form categories. This one does though. This one, Xanthoparmelia cumberlandii. This is a clear foliose species that also grows on rock, growing not far from where the Rhizoplaca subdiscrepans was. You see it has a blue-gray thallus with fine lobes kind of stick up off of the substrate a little bit. Doesn't necessarily, you can find it more oppressed. And then you've got these big brown apothecia. We've got 4 common species in the Xanthoparmelia genus in New Jersey. 2 of them, all 4 grow on rock, or more abundantly in North Jersey than in the Pine Barrens. 2 of the species have apothecia. This being one of them, the other being Angustophylla, if I remember correctly. And then 2 species are isidiate. They have isidia and reproduce asexually, and they rarely to never have apothecia. And then of those 2, in each category, apotheciate or isidiate, one species has a black underside and one species has a pale to light brown underside. So if it's got apothecia and has a pale to light brown underside, like this, it's Cumberlandia. If it's apotheciate and has a black underside, then it's Angustophylla. If it's isidiate, then it's either Plidii or Conspersa. I believe Plidii has the pale underside, and Conspersa has the black underside. I'm gonna have that backwards, but basically, in order to do these things, you got to know what the reproductive structure is and what the color of the underside is. And between those 2 characters, you can narrow it down to one of the 4 common species in New Jersey. Next up, we've got this guy from South Jersey. You can see it's last week we talked about blue-gray oppressed species. This has the squared shape to the thallus, and it's got that hammered, wrinkled appearance, making it Parmelia squarosa, but it's discolored and brown instead of blue-gray. And if you look closely, it has these little orange growths on the thallus that are super tiny. So this is a Lichenicolous parasite that is stealing resources off of the Parmelia squarosa. And I believe this is Marchandiomyces corallinus, which it does not appear on the current lichen checklist. So this doesn't appear to be super common in New Jersey, or at least not commonly recorded. I tried to do some microscopy, but it didn't really tell me a whole lot.
What are the orange?
So those are the fruiting bodies of the parasitic fungus that is attacking the lichen host, Parmelia squarosa.
That's excellent. I mean, probably not for the lichen, but
Yeah. But, the Parmelia squarosa is super common. There was a lot of it at this site, and not a lot of the Marchandiomyces. So the population had enough Parmelia to spare for this little Lichenicolous guy.
So are those little fruit bodies of the parasitic fungus, are they like less than 1 millimeter, I guess?
Oh yeah, yeah,
Wow.
definitely. I mean, let's see if I have a folder here. Yeah, I mean, I'd say, the width of these lobes here is, probably half a centimeter at most. And then lastly, I forget if I've shared this one already at Taxonomy Tuesday, but worth sharing again. This is Schismatomma glaucescens, which are these little white circular apothecia growing on this bark. This is a species that grows apparently exclusively on oak, and it grows in the ridges of the oak in the pine barrens. So it's not easy to find unless you've seen it before and you kind of know where to look. It doesn't appear to be super common, but I suspect that it's more abundant than the records suggest, and it just gets overlooked. I'm guessing it grows primarily on scarlet oak and maybe some of the larger, uh, post oak. I'm not sure. I would expect to see it on, white oak or some of the bark that's a little bit flakier and blockier rather than having these long longitudinal ridges. But uh, this is the only observation of this lichen species on iNaturalist from anywhere in the world, which is kind of cool. So
The only one
You're
on
it?
the entire world.
on
Wow.
iNaturalist, yeah, I mean,
Wow.
there's collections in herbaria and stuff, but iNaturalist, this is it. So something to keep an eye on for out for in the pine barrens. If you see this type of oak tree, look closely and see if you see these little white apothecia, because this is probably Schismatomma blackescens.
Huh.
What type
And that's all I got.
of oak is that?
Say that
Which
again.
which oak?
This one I think is the scarlet oak, is Coccinia.
Oh, okay. I don't think I get those. Cool. That's so cool.
Could we get a zoom in on that one again?
Yeah, this one's probably the best.
Man, you're up
So,
close on this, huh?
yep. I
So
always like to tell people if you like sticking your nose in places, you might make a good lichenologist.
so I imagine these probably like most lichen fruit bodies will persist through the winter, so you can
Yeah,
probably find them in the winter.
yep,
Yeah.
definitely.
So those are also probably they were even smaller than the other things, I would say, or about the same.
Um, I'd say these are larger. These are
These are
larger
larger.
than the
Oh, okay,
Marchandiomyces. Yeah.
okay.
I spotted these with the naked eye. Um, you can get a lot, you can see it a lot better with the loop, but they're, if you know what to look for and you know the microhabitat, it definitely helps.
And they're not exclusive to that one kind of oak. It's just oak in general.
Apparently they like oak, but not all oaks have the same type of bark. So I mean, it's hard to tell, but I've found it on scarlet oak and that kind of scene. The habitat says in the ridges of bark on oaks, and not all oaks have ridges. So
Yeah, because even the willow oak in my yard, it's got some pretty deep.
yeah, that might be one
I'll
to look for it on.
have to stare closely at it. Thank you, Jason. From a hunter's thing before, I was looking at the genus, those Xylobolus, because I was looking at the other ones like, oh, well, what do they look like? And they're so different than that.
Yes, if you scroll up and click on species instead of observation, you can see one thumbnail of each of the species. Yeah, see where, yeah, right there.
Oh, weird.
There's four there, aren't there? Four.
Yeah, when I looked, I filtered on Eastern North America. This looks like it's
Uh,
searching
Oh
okay.
globally.
man.
Okay. Mm-hmm. The third one looked a lot like something else to me. Like that looks like,
Durium.
yeah, complicatum or the other one, doesn't it? This, yeah,
Yeah, I'd probably just pass that by and think it was Stereo.
yeah, me too. But then the others don't even look the same. Oh,
Oh, okay. That photo looks different.
yeah.
So this uh, after J or I, is it I or J? Oh, they're all I. The one right close to the middle, two of them close to the middle. It looks like they're labeling them as spectabulus, but they look like subpileatus.
Yeah,
I
they do
wonder
to me
how you
too.
would tell the difference.
Right, especially when the ones on I mean, the ones on the left look very different, but yeah,
Yeah,
some of them. Hmm.
I don't know these. The only one I know is frustratulus.
Yeah, I love that.
That one's pretty easy to identify in the field.
Super. Yeah, that's a good one. Easy to know.
I can't say that I know much any of this.
Look at some more pictures of this one because it's it's often on the ends of cut logs, of cut wood, and the second on the top, that's really typical. Like that, yeah. You'll start seeing it everywhere now. It likes oak. Um, I don't. I think it might grow on other stuff too, but yeah, just like old logs that have been cut. If you look at the cut end, often you'll see it. Ceramic parchment. Yeah, it reminds me of tiles or like a mosaic a little bit.
Yeah, it
Yeah,
usually grows on very, very hard oak, so it's really difficult to get a piece of it off to take home and study.
it seems pretty variable as well in
Some of
the
that
color.
variation is probably by age.
I think everything that looks like subpileatus is subpileatus, and spectabulus is restricted to the Indo-Pacific and China, and the stuff that's labeled as spectabulus in North America is likely all subpileatus.
Oh, interesting.
Okay, well that
What if,
sounds good. That would
and
mean that those.
oh,
Oh, go ahead.
no, you go ahead. I'm sorry.
No, that's all right. I was just going to say that would mean that there's probably not a big potential for confusion then between those two species. That would be good.
So the Principus or whatever, that's also not in Northeastern America.
Right. Yeah. Looks like the type specimens from
Huh,
West Java.
look at that.
From where?
West Java,
Oh, wow. Okay. Cool.
Indonesia.
Yeah.
Some Maricel on that one.
Oh, nice.
Yeah, I guess those are cystidia. I'm guessing.
Hmm. Well, I guess this is some more stuff to look for in the winter.
It is.
Probably persists.
Yes. It is interesting to see how, like, as we look at these INAT pictures, how there's quite a few that I'm like questionable, like, wait, it looked like there was that Krishnamaria. Someone put that in one of those.
Hmm.
Oh, this one's sort of similar to Frustulatus, a little.
There's no observations, no identifications.
None. Hmm.
Hmm. Hmm.
Interesting.
Any anybody else got some finds? They don't have to be fresh.
I could throw one more in if nobody else has anything. I had thought I included it so but if there's anyone else,
Go ahead.
okay, let's see. I gotta figure out how to do this though. Um so yeah, I'll try. Oh, I remember now, sorry. So I'll try just putting it in the chat instead of emailing it, but it's an INAT observation.
Cool.
Okay. And I thought this was really cool, but I don't know. We'll see what you guys think. So this was growing on a log. Are you gonna open it, Chris? Can you
Yeah,
open?
yeah.
Okay. Um, down here in Louisiana, and when I touched it, it was kind of puffy, almost like a puffball, and I also thought it reminded me of some of the gyrometras, and I really didn't know what it was. And so then I collected it, and
Ooh.
this is the underside that was attached to the log, and it kind of seems to be wanting to make gill-like structures, but of course, but there are no true gills, and actually, I think the spore-bearing surface is on the cap, um, and it's trichoderma. So and you guys, I'm sure, know that is the green mold that will show up on your sawdust blacks if you're growing stuff, growing mushrooms, and you get that mold. That's trichoderma, but this is sort of a different manifestation of it or something. But these, I actually did the microscopy with some help. I got a lot of microscopy time down there. I brought my microscope. It worked out pretty well. But there are the asci here, and they're very tiny, and it's not great. But yeah, that one's pretty good up there. And so yeah, have you seen that before?
I don't think I've ever seen a trichoderma that large, but that's
It was a big one. I did find a smaller one on a different log in the same park, like a couple days later. Um, yeah, pretty interesting.
so are these like uh just the singles are round in there?
That's a good question.
This,
Yeah, I mean they appear to be pretty big. I mean, I guess pretty small, but I don't know. I wonder if this was I used immersion to see one thing, but I don't think this was it. Yeah, so that was a new one for me and pretty interesting. I have it, or the other one, so I can maybe do a little more work on it. But yeah, pretty weird.
And you thought the spore-bearing surface is the top?
Yeah, that we scraped off the cap cuticle I'm pretty sure to get that microscopy.
That's interesting.
Yeah,
Yeah,
this reminds me of that. I forget the name of it. It grows in where there's been a fire in like a spruce forest. I think it starts with R.
Yeah,
But
what's
that,
that called?
oh, what's that called? Yeah, but that the spore bearing surface is just the upper surface,
Hmm.
Hmm.
and
That
and
might
this
be
thing
cool
too.
if you still have it to like section it so you get a, some of that outer surface in the layer that you can like zoom a couple of times down,
yeah,
just cut like a thin as slice as you can, like through a bunch of the material that way.
yeah, to see kind of the contrast and um yeah, it's funny
Dave,
as go ahead, Jason. What?
Dave, you're thinking of Rhizina undulata.
Yeah, that's it. Rhizina. Yep. This is similar to these, but those grow on the ground that's been burned.
Yeah.
And I've seen this thing before too, and I forget what it was called when I saw it. It might be a different name years back.
Trichoderma is a really interesting genus. There's some really bizarre diversity. It's like I'm looking on INET. There's like upwards of 35 or 40 species in Eastern North America.
Wow.
Some of them are club shaped. Some of them are like this one. There's the little green moldy thing. Um,
That
this,
yellow thing that we find once in a while.
mhm. And then there's also there's a species of ant that grows in the pine barrens and elsewhere that um, farms fungi underground, uh, and they
Mhm.
Trachymyrmex septentrionalis, and apparently the fungus that they farm is something in the genus Trichoderma.
Hmm.
Interesting.
Are those mound ants?
Um, let's see. I think they do make mounds in the sand. Let's see.
Which, for the record, don't stand on one and try to look at all the little holes because then they'll start crawling on you and biting you.
Yeah, like little crater type things.
Yeah, I found it. This used to be Hypocrea peltata.
Ah,
Yeah, yeah, that was the name I knew it by.
interesting.
There's probably a lot of old genera that have been lumped into trichoderma based on DNA. I mean, what Jason is saying is there's this really weird diversity of morphology, which implies to me that probably there were different genera for some of these things just based on what they looked like. Um, so I imagine there's a lot of times DNA results in a lot of splitting, but sometimes you get a lot of lumping because you get these genera that just have ridiculous morphological variation.
Yeah, right. That's what's so interesting. So how many members are there in Trichoderma? Jason, do you see uh, about how big it
It
is?
In eastern North America, I see 38 species.
Wow.
And let's see, total. Yeah, here, I'll share my screen. Um,
Thank you.
yeah, I mean, look at all these things. It's just like,
Oh, wow.
yeah, let me review this guy.
Actually, I'm looking in the Bisset ASCO book, and what they used to call hypocrium was also weirdly variable morphologically. So,
Yeah, I see 69 species total on INET. Um, 37 or 38 in eastern North America. What's this guy doing out there? So, yeah, here's the peltatum. Here's the Verity that we see a lot. Here's that club-shaped gills.
Wow. Yeah, to see these club-like ones is very interesting.
Yeah,
That's
Holy,
just a straight gilled mushroom. That is crazy.
yep.
the
Yeah,
one,
this almost looks like a slime mold. Some,
yes.
I just looked on Michael Quebec, and you can put a genus name into a field, and it'll show you all that it has in that genus on Michael Quebec, but it also shows you um, sometimes other things that used to be that in that genus, but now they're in a different genus. And I did a search on where, let me see, let me go back to where was I? Oh yeah, on trichoderma, and actually I did a search on. I'm sorry, the other way around. I did a search on hypocrea, and I'm getting a whole bunch of trichodermas,
Oh,
and one protocrea, which I don't know what that thing is. But there's all these trichodermas, and they look really weirdly variable, like Jason's saying. So it
yeah,
looks like trichoderma inherited quite a few of these hypocrea species, and probably some other things as well. So it's kind of interesting. I wonder if these are all teleomorphs, or some of them are anamorphs, or you'd have
mm,
to do a little bit of reading to try to
yeah.
get a handle on that.
can you review what those mean again?
Well, some fungi occur in two stages. An asexual stage, I believe that's the one called anamorph, and it produces conidia. Usually, they produce conidia, which are like spores, but they're not sexual spores. And then the same genetically um, uh, identical species occurs in a different form called a teleomorph. Um, and it's a lot of times it's suspected that there will be an anamorph of some fungus, but they don't know what it is. And
Mhm.
it gets even more weird. There's also endophytic fungi that like live inside plant cells that are
Yeah.
now they now think that some of those manifest also as saprobic fungi
Hmm.
at different stages in their life cycle. It's there's really a lot to be understood about this.
So the canidia look like the other spores, but they're not able to reproduce.
Now they look different. Usually the canidia are usually small and round, and for the most part, I mean, I'm sure there are some exceptions, and variable in shape, in size,
Oh,
and the ascospores, the sexual spores are those are usually more distinctively shaped and ornamented, and that they have like more character, than the conidiospores.
okay.
And the conidiospores are not born in asci. they're just like linked
Oh,
together and stuck to like hyphae or so. I don't really know. I'm just guessing at this point. Um,
well, that
yeah,
helps.
there's a lot to understand there, and most of it I don't know.
Thank you.
I got anamorph refers to the sexual stage of a fungus, like a mold. A
Oh, do I have it backwards? Is teleomorph the
No,
asexual?
no,
oh, the asexual
Uh,
stage of a fungus.
yeah, anamorph A, asexual A.
yes. Okay. Thank you.
That's what you had said. Yeah.
And then, yeah, teleomorph is the sexually reproducing stage of
I
a
wonder,
fungus producing sexual spores like ascospores or basidiospores through meiosis.
And then the holomorph is the entire fungus that includes both the anamorph and the teleomorph.
Hmm.
cool. And you can see them side by side sometimes, right?
Ooh.
Um,
Or
well,
no?
usually they have alternate hosts, I thought,
Oh, mm-hmm.
and possibly alternate seasons. Like the anamorph is of one time and the teleomorph is another. I could be wrong about that. I know that's the case in some. I don't know if that's universal. Um,
Okay, cool.
Nice. Anybody else got anything? We got there's a bunch of people in here. Bianca's hiding some mushrooms maybe, or Shihan.
I haven't even seen mushrooms. I don't get out in the daytime anymore.
I could throw another in if you want.
Sure,
Got one I can show. Whoops, sorry.
go for it.
Go ahead, Hunter.
No, you go ahead. Is that Tristan?
No, it's Paul.
Oh, you should go.
All right, thanks. Let me share. I think I've got something applicable to what we were just talking about. That's
Oh,
it.
good.
Um, this one here. Let me get this out of the way here. This little white blob fungus here,
Yeah,
growing on. You know
yes.
this one?
Yeah, I love it. Isn't that the white blob? That's a joke, right? Like, remember? Yeah, yeah.
Yeah.
I love
Yes.
one.
So most of the
Actually,
time, there's a species name attached to this. Also, compressive something.
right.
Um,
I noticed that there was about 20 different species for this
oh,
Helicogloia. Um, iNaturalist certainly does want to suggest compressa, but I'm a little leery sometimes of. But I think this is one of those perhaps anamorphic stages we're talking about. I do have some micro photos, but yeah, these little gelatinous but firm white blobs. Um, and
You
I think
think that's
this,
the animal?
I think it is, from what I've been reading, and I've got this picture which shows this sort of, uh, it almost looks like an aquatic vegetation or something here. These strands, but the next picture I think is what's showing the the conidia being formed on the ends of these on the ends of these branches here. So I think that's what we're looking at here.
I'm going to look it up in the ASCO book, but I think you're right. This, I mean,
This is a,
photos I've seen
this
of
is a
conidia.
basidium. From what I understand, it's a basidiomycete. This
Oh, it might not be in here. Okay.
right.
I thought it was, I'm probably wrong then. I thought it was an ASCO. I guess I'm just,
From what
I've
I've
got,
read, I think it's a basidio. Go
yeah,
ahead.
okay, yeah. I've got observations of it on Mushroom Observer, and I know I've found some what I thought were spores. Let me look and see what.
Yeah.
Yeah, if
For
these look like conidia, and if
Wow,
they are, then they're asexual, and if that's the case, then that would make this an anamorph.
that's awesome.
Yeah, the description for compressa talks about slightly compressed branches. That's I guess sort of a relative thing. So I don't know whether this exhibits that characteristic, and whether I should call this species compressa or just leave it for what it is right now. But yeah, what I did was I just sort of cut the very tip off of this and with a razor blade and got it squashed under a cover slip. Um, but anyway, it's growing on hardwood. Probably a tulip, maybe an oak.
That's great microscopy. You must have
Uh,
been excited to see that underneath there.
I was, yeah. I'm just sort of learning about using some of the dyes. This is a dilute Congo red that I'm only just starting to practice with, and that I think showed things up nicely here.
Very
Um,
nicely. What about the other one? What were you using for the
this well, this one is. I think that this was without any sort of dye. My digital camera tends to give things sort of a green
okay.
cast. It's some sort of an anomaly. That's not. It's really not green, but
Okay.
this was just in water.
I was curious. Yeah.
Yeah.
But
Oh,
yeah, the Congo red images really look nice.
I just
yeah.
put a link from Michael Quebec to Compressa into the chat, and the micro they show looks just like what we're seeing here.
Okay.
And yeah,
Yeah.
this is an anamorph of some Basidiomycete. So I was wrong. I thought it might have been an ASCO, but it's not. Um,
Yeah.
but it doesn't say what it's an anamorph of. So it might be one
Hmm.
of those mysterious things.
Hmm. You mean
Yeah.
some species other than compressa?
Well, it would be the same species, but it might have
Okay. Oh,
traditionally have had a different name because the teleomorph might
oh,
just the link has not been discovered.
I
Um,
see.
yeah, there are examples, I believe, that still exist of um, fungi that one stage is known and the other isn't. Presumably,
Hmm.
any anamorph is going to have a teleomorphic stage as well, but um,
Right.
in this one, it looks like it might not be known. I'm going to look again in the remarks. It just says this tiny anamorph is characterized by production of conidia on conidiophores, right, with
Mhm.
slightly compressed branches. Okay,
Right.
like you were just describing,
Right.
and it
Right.
doesn't say anything about a teleomorph.
Well,
Um,
it's not
let
a
me look this.
necessity to make a mushroom. I mean, for a mushroom to exist, a sexual stage is not a necessity, right? So it conceivably not have one.
Yeah, that's true. They're saying now that Amanita phalloides reproduces asexually. So I'm not sure how that works, but I've just heard this recently. But
Mhm.
anyway, I'm going to do a little search here on
But that would also mean that,
Telium.
like, when we look at the spores of an asexual, like, we're not really seeing the, what we think of as real spores then, right? It's just conidia.
Yeah.
Yeah.
Yeah, I'm mystified by this Amanita phallides thing. I'm not sure exactly what is meant by that. But let me see what I come up with here from this search. I'm searching Heliocogloia compressa teleomorph. Let's see if something comes up here.
Up until recently, it might have been called, this genus might have been Leucogloia.
That's what
Well,
I'm
not,
seeing here. Yeah,
yeah,
Helio
okay.
Facebook mushroom identification. Okay, there's some pretty good people on some of these Facebook pages. An imperfect fungus that has no. Oh, um, an imperfect fungus that has no fruiting body. But that looks these globs look like fruiting bodies to me. Um,
Well, the anamorph has a fruiting body.
Hmm.
Are they saying the teleomorph does not?
they're not even using the word teleomorph.
I don't
I
think
think
the
they're
teleomorph is
considering these
known.
globs to be not fruiting bodies. That doesn't seem right to
Hmm.
me. Um,
Hmm.
hmm.
Yeah, puzzle.
Fascinating.
Well, in in plants, the fruit is the reproductive structure that is a result of sexual reproduction. So if this is an anamorph, then it's possible that no anamorphs are considered fruit or fruiting bodies if
Mhm.
they're asexual.
I see.
Hmm.
Okay, I got
Okay.
it.
Yeah.
Yep,
Okay.
that makes sense. Thank you.
Yeah. Yeah, that makes sense.
Oh, yeah. Sure
And
does.
then, so yeah, I think Hunter's right that, not every fungus needs a teleomorph, but if this does have a teleomorph that's just unknown and it's, it could have a completely different form and be completely, you know, no conceivable way to know that it's in any way connected to the Helicoballoa. But
Except maybe for DNA.
right, except for DNA. And then at that point, if the connection is found, presumably we'd have two names for the same fungus, and whichever name was described first would be used to describe the whole thing, right?
That would be the rule, yes. And that has, I believe, happened a few times
Yeah.
where a teleomorph and an anamorph have been found via sequencing to represent the same biological species, genetic species, and then they have to change the name of one of them according to the rules.
That's cool. That's DNA's really helping in that area. That's fascinating.
Just maybe I'll do one more. Let's see. This one I'm not sure. I think I've got a tentative genus on this of Gymnopus. Um, these caps are about a half an inch in diameter. Um,
Oh, we've been
closer
seeing
here.
a lot of these.
Okay. So I've got some spores there, which somewhat
Nice.
ellipsoid, somewhat tear-shaped. That could be consistent with Gymnopus, I think. But just wondering, whoops, very red at the base of the stem.
Yeah, what's that? Were these on wood?
Um, Gosh, hard to say. Sort of emerging from
Oh,
the leaf
okay.
litter.
Yeah.
Hard to say exactly. I think it was just coming from the leaf litter. Oops.
So there's a gymnopus that has a red stripe like this. I think it's maybe a erythropus or something like that. Let me see if
Yeah,
I
I
can find
think
it.
Brandon showed these a couple weeks ago, or somebody else. I
Well,
think.
I had something that I wasn't sure if it was the
Oh,
gymnopus or the conopus. No, I don't think these are the conopus. Um, Acervitus, I think, Conopus acervitus, but I don't think that's what these are. Look at the bottoms of the stalks, though. It looks like there's something stuck to them. Oh, are these hairy on the bottom? Are these stalks hairy? Oh, they're pretty hairy.
Oh, yeah, that could be. Yeah, it could be
So
some.
one possibility would be what is it? Spongiosis, I think, is one of the species, but that usually has a thicker stalk. Um, Gymnopus makes sense here, though, and in
Okay,
in the strict sense, not
right.
Colybiopsis or Marasmiellus, some of the split off genera.
Okay,
Although, but
yeah.
those are Marasmiellus is or Colybiopsis, I guess, is a possibility, but these look more like real Gymnopus. Let me
the,
find
the spongiosis
Erythropus.
spores look really different than yours. Can you?
Oh, okay,
Yeah,
okay.
I don't think it's that. They're really sharp. Yeah, they're really sharp at the ends on Mushroom Expert at least.
Okay.
Yeah, a lot of the Gymnopus spores are are really almost like fusoid, meaning really tapered and pointed, like you're saying, Hunter. Let me
Look at
look up see what I can
look at the
find.
way the some of these are. You can see like if you catch them in the right, you know,
Oh, they are. Look at the one that upper
top
middle.
right,
Yeah, top right. There's
straight down
yeah,
from
those are
that one.
yeah, yeah. But it was said though some of them are tear shaped. So yeah, a lot of the Gymnopus spores a lot of times depend on whether you see that depends on how it's lying down in the mount.
About Gymnopus erythropus.
Okay.
Yeah, that's the one that I was thinking. What do the spores look like from that?
Yeah, I'm looking for a good spore photo. I've got one that's decent. And look, it's not unlike this one.
Been about 8.
Michael Quebec has a nice. Let's see, is it it's a drawing, but it looks like these spores. Let me get a link for that and paste it in because I think that's what this is. These spores look like what I'm seeing in the drawing.
Yeah, morphologically too, for sure. They look very similar.
Yeah, yeah. I think we may have nailed this one. Oh, great,
Erythropus.
Rithwipus. Yeah. It's got a red
Okay.
stalk like these do.
Okay. Yeah, I thought the stem was pretty indicative of something. It was fairly red in color.
Yeah, I just
I
put
think
into the link or into the chat a link
okay.
to Gymnopus erythropus, and you
Yeah,
can
those
see that
are
the spores really look pretty similar.
Guess this is a basidium.
Oh wow, what a nice photo of a basidium. Look at that.
oh. So there were probably four spores on top of there, but now there's just the two.
Now it looks like I'm
Yes.
seeing four. Well, I'm seeing four sterigmata.
Yeah,
There's
okay.
one in the forefront that's kind of pointed and a little bit hard to see. There's one in the
Mhm.
back that has sort of a nice round sort of
Mm.
thing where the spore had been attached, I guess. And then there's one up above and one below. Looks like there's four of them.
Yeah, four sterigmata, but yeah. Yeah,
Yes,
cool.
those are Yeah, so those must be spores under development there, those small.
I don't know if those are just where there
Okay,
used to be spores attached or if
okay,
those are immature spores. My guess is those are where spores used to be attached.
okay.
I'm not sure. I don't know.
Yeah,
I have no idea, honestly.
yeah,
I'm guessing.
sure.
Hmm,
Yeah.
interesting.
Okay. Well, that's good. Yeah, I'll do a little more reading up on Erythropus and maybe we'll call it that. All right. I appreciate the help as always. I'll stop sharing.
Nice
Thanks,
photos.
everybody.
Yeah, that was a really good observation.
Really nice.
Could we see the photo of the gill edge again, or the part of the gill? Let's see if there's any of those cystidia there, because it's got that species has really weird cystidia.
You mean here?
No, the micro.
Oh,
The
sorry.
Yeah, the one with the
Oh, yes.
bastidium. Yeah,
Yeah.
here. Yeah, okay. Yeah, if they're in there, then they're pretty well obscured. Okay, I
Yeah.
just wondered if we might pick one of those out. Sometimes these cystidia are really hard to find because they're like hidden in the gill trauma. I'm guessing that's what's going on here.
Okay. All righty.
Yeah, that's why I've been trying to get like kind of like a neat photo of a gill edge, and then I'll go back in and chop it up to try to just break stuff loose.
Yeah, sometimes I smash my mount like extra hard. As long as there's no like debris under the slip
Right.
that's gonna shatter the slip. And sometimes, right, like you're saying, Chris, sometimes you can free up some of the cystidia, and that seems to be more likely in some genera than others, where you can free them up, and then they're sort of like free floating in your liquid mounting liquid.
The other thing I've been going back to doing a little bit is I kind of like I don't know, I'm trying to go in like a bit of an order. Like I'll try to do like a real like Roman aqueduct type of cut to see if there's just anything jumping out at me like in a specific place. I just run over it real quick and then I'll chop it down further.
Well, that's probably the recommended approach if you are patient enough to perform this Roman aqueduct thing, which just really gets on my nerves and results in having a lot of razor blades get thrown away, or at least demoted to do something other than try to make a Roman aqueduct. But that's a great strategy. If you can get those a good Roman aqueduct section, then as you're saying, you can see where things are happening, and then maybe try to zero in on those areas, or maybe smash and look again in those areas. That sounds
Well, a
like a
lot of
really good
times,
strategy.
even if you don't get a good one and you only get like, let's say, like one gill falls off, but it falls off in a thin enough long slice, a top to bottom slice, then you still got what you wanted.
That's a pretty good idea also, because then there's sort of less clutter. If you just have
Exactly.
like one strip of gill, I can see where that would, yeah, that if you can, once again, though, you have to be able to make these really, really thin sections. And
Well, what I usually do is like even like a you could even take just section out a whole gill and do the exact same thing. Just cut a chunk away, and then a lot of times if you take like another clean slide and just put it use it like use it as like a little cutting edge and you could just slide that over a little bit and then cut right along your slide. Sure.
Oh,
And
I see. So yeah, so it's like you're using the slide like a straight edge kind of just to
exactly.
yeah. Well, huh, oh, those are good ideas.
Yeah,
People are
because
wondering,
honestly, I don't know if I find any extra anything, but I feel like there's a better shot.
You know the orientation with the aqueduct, which is nice.
Yeah, that's the big advantage with that. You know where the edge is, you know where the face is. That's why it's the recommended way to go.
Well, I think I can show this other one if you want. But I have to go after that, or we can wait till next week, or if someone else has something. But I should get going one way or another.
It's your call whether you want to show it or not.
Okay. I'll show it quick. It is. I just want. I guess I just kind of want confirmation. From Dave or Igor or any of you. That it is what I thought it was. I wasn't able to do microscopy on it. It's in the chat now. The INAT observation, but I don't have this Amanita in Illinois, but I believe it's pretty common down south, and you guys might have it too, if I have it correct. I believe it's Polyparamus.
The pine barren species and a coastal New Jersey species, and it looks like I mean Igor probably can tell you best, but it looks like when I've been in the South Jersey, South Central Jersey, closer to the coast, this is where I found this species, and this is what it looks like. What do you
Oh,
think, Igor? Polyparamus?
I don't know if he's here still.
Okay.
he's here.
I know they're supposed to have they can get quite large, and then there's like warts on the cap, which there were, and then a lot of material on the stem.
Yeah, polyparamus means lots of pyramids, and so that's what you have. You have these scales all over it that are like little pyramids, and they tend to lose their rings. So this did have a partial veil, but it's not persistent. I I think you're right. I think that's what this is.
Okay, cool.
Look at the gills are. Is that just shadowy, or are they actually a little bit sort of like beige? Because there are these lepidellas that I know Rod Tillis would describe the gill color as cafe au lait, coffee with
Mm.
a lot of cream in it.
Yeah, I noticed in some of the pictures, they almost look gray or something, but this looks more cafe au lait.
Yeah, let's
Uh,
see. This one might be on Mushroom Expert because he covers species that go into the south. Let's see if that's on.
I mean, you got plenty of pine needles.
yeah, yeah. And then the gills are forking at the edge. Is that right?
Or are they just like
Stuff
this, like
together.
they're all like stuck together? Like it looks like you got like three like on that one.
Yeah. Okay.
Yeah, those gills are probably just stuck together.
Yeah,
Yeah,
same as
this
that.
species is on Mushroom Expert, and Michael Quo describes the gills as creamy or dirty buff.
Yeah.
Okay. Yeah. There we go.
So yeah, so right,
Cool.
nice, good,
Well, that's
good
it.
one.
Please. Yeah. Good to get this down. All right. Well, I'm gonna run, but I can't wait till next week. Have a great week, everyone. Bye.
Bye
Thanks,
bye.
Hunter.
Bye.
Thank you.
Bye, Hunter.
Well, if nobody else has got anything to show, maybe we call this one this evening.
That's
Yeah, if
fair.
I get out
Screw us.
cross country skiing, maybe I'll find some lichen.
Nice. All right, guys.
Yeah, thanks for hosting, Chris.
No
Thank
problem.
you.
Thanks, Chris.